Overview of analytes, non-protein nitrogen. So non-protein nitrogen is traditionally used to monitor renal function or kidney function. The term originated when analytic methodology required removal of protein from the sample before analysis. If you recall, proteins and amino acids consist of that N-terminal amino group, so that can interfere with nitrogen concentrations and measurements. So the concentration of nitrogen-containing compounds was quantified spectrophotometrically by converting nitrogen to ammonia. A subsequent reaction with Nessler's reagent produced a yellow color. The NPN fraction comprises about 15 compounds of clinical interest. The majority of these compounds arise from the catabolism of proteins and nucleic acids. Urea is probably one of the most frequently ordered tests, so this is going to be on a lot of your basic metabolic panels and basic lab draws. Urea is the major excretory product of protein metabolism and is present in highest concentrations in the blood. Biochemistry, protein metabolism produces amino acids that can be oxidized to produce energy or stored as fat and glycogen. During protein metabolism, nitrogen is released and then converted to urea, excreted as a waste product. Clinical application, it is used to evaluate renal function, to assess hydration status, to determine the nitrogen balance, and to aid in the diagnosis of renal disease and verify adequacy of dialysis. Analytical methods, the most common method couples the urease reaction with glutamate dehydrogenase, where the conversion of nicotinamide adenine dinucleotide reduced NADH at 340 nanometers is measured. So spectrophotometric assay. Uric acid. Uric acid is formed in the liver as the end product of purine metabolism. So if you remember DNA nucleotides, we have purines and pyrimidines. The purines are adenine and guanine, A and G, either through the breakdown of dietary nucleic acids or from cellular turnover. Reabsorption of 98 to 100% of the uric acid from the glomerulofiltrate occurs in the proximal tubules. Nearly all of the uric acid in plasma is present as monosodium urate. Clinical application, it's usually measured to confirm the diagnosis and monitor the treatment of gout. Assess and prevent uric acid nephropathy during chemotherapeutic treatment, to assess inherited disorders of purine metabolism, to detect kidney dysfunction, and to assist in the diagnosis of renal calculi. Methods, so these are measured enzymatically. So uricase, the enzyme that catalyzes the oxidation of uric acid to allantoin, are more specific and used almost exclusively in clinical laboratories. Coupled enzymatic methods measure uric acid levels by measuring the hydrogen peroxide produced as uric acid is converted to allantoin. So again, increased plasma uric acid concentration are seen in a variety of clinical conditions, include gout, increased catabolism of nucleic acids, and renal disease. Creatinine and creatine. So creatine is synthesized primarily in the liver from arginine glycine and methionine. It's then transported to other tissues such as muscle, where it is converted to creatine phosphate. Creatine phosphate is used as an energy store, so it can easily donate a phosphate to ADP to make ATP. Adenosine diphosphate, when you add a phosphate group to it, it can create ATP or adenosine triphosphate, and then that can be used for energy in muscles. This is a short-term energy fix, so the more creatine, that phosphate present, the longer you will have that energy fix, but this is not like life-sustaining energy. It's just a short intermediary energy source. Creatinine is released into circulation at a relatively constant rate. that has been shown to be proportional to an individual's muscle mass. So clinical application, plasma creatinine concentration is a function of relative muscle mass, the rate of creatine turnover, and renal function. So creatinine clearance, a measurement of the amount of creatinine eliminated from the blood by the kidneys. Creatinine clearance and glomerular filtration rate, or GFR, are used to gauge renal function. The GFR is the volume of plasma filtered by the glomerulus per unit of time. So creatinine is measured using either the Jaffe reaction or a kinetic Jaffe reaction. The initial Jaffe reaction has kind of been phased out, but it's when creatinine reacts with picric acid in an alkaline solution to form a red-orange chromogen. That chromogen can then be evaluated using a spectrophotometer. The kinetic Jaffe method deals with the reaction with various enzymes, and it increases the specificity of the assay method for creatinine. For creatine, the traditional method for creatine measurement relies on the analysis of the sample using an endpoint Jaffe method for creatinine before and after it's heated in an acid solution. Creatine can then be measured by HPLC, high performance liquid chromatography. So it may be measured in plasma, serum, or urine. Hemolyzed and icteric samples should be avoided. May be refrigerated for 4 days. If longer than 4 days, the specimen should be frozen. Sources of error are listed below. Bilirubin causes negative bias in both Jaffe and enzymatic reactions. The patient use of certain types of drugs should also be noted. And then here's some reference intervals for the various plasma or serum creatinine, and then in the urine. Ammonia. So ammonia is produced in the catabolism of amino acids and by bacterial metabolism in the lumen of the intestine. Ammonia is excreted as ammonium ion by the kidney and acts to buffer urine. Clinical conditions in which blood ammonia concentration provides useful information are hepatic failure, Ray's syndrome, and inherited deficiencies of the urea cycle enzymes. Ray's syndrome occurs most commonly in children. It is of use in the diagnosis of inherited deficiency of urea cycle enzymes. So analytical methods for evaluating ammonia. Accurate laboratory measurement of ammonia in plasma is complicated by its low concentration, instability, and pervasive contamination. Current assays usually detect either NH3 or NH4, ammonium or ammonia. Can be measured by enzymatic method using glutamate dehydrogenase. This method is convenient and the most common technique that's used currently. So high concentrations of ammonia are neurotoxic and often associated with encephalopathy. Encephalopathy refers to brain diseases. Ammonia alters metabolic processes in the brain, which results in accumulation of toxic species and impairs astrocyte function. So astrocytes are one of the glial cells that support the brain and protect that blood-brain barrier.